Hi Federico.
Hello, Sonia.
Hello. Hi, could you tell us a little bit more about your research and what have you learned during this time in the lab?
Yeah, sure. So I'm basically a physiologist, and I study how the distribution of synapses over a dendritic tree in pyramidal neurons influences neuronal output. To put it a bit more simply, neurons have really large dendritic trees, and they receive thousands of synaptic inputs. And also these dendrites, we know that they taper towards the tips. So they start very thick, close to the soma, and they become thinner as you move towards the tip of this dendrite. Now this has important implications in terms of electrical resistance. So if you imagine a tube becoming smaller, you can see how less water will flow through. So it increases the resistance in the same way with electrical currents.
Yeah.
And when you have increased resistance, with a similar amount of synaptic current, you would have a larger membrane depolarization. And what that equates to is a larger synaptic signal. So the neuron needs strategies to normalize these synaptic inputs and make them more equal along the whole length of the dendrite. So what it does, it reduces the size and the strength of synaptic inputs as you move from the soma towards the tip. So you get larger synapses proximally and smaller ones distally. So we actually were able to confirm this in the lab, and this is just being published last month as part of a larger study. But they say part of these results were regarding exactly this. So dendrites become thinner, and you have smaller postsynaptic spines on these dendrites. So the natural question that came to us was, what do the presynaptic inputs look like? Because we know that they should match the strength and size of the postsynaptic inputs. And this in turn will have a huge effect on how these synapses behave, because if you have a smaller presynaptic side, you're not just reducing the strength of the signal, you're changing the way it's responding to the action potentials coming in, because you're going to have with smaller Burrone, you're going to have less release sites, and your release probability should go down. So basically the likelihood to synapse responding to one action potential. And with that, you're going to change its properties in terms of plasticity. So what we set out to do was to look at the structure of these presynaptic inputs and also their function. So first of all, what we did was some 3D EM reconstructions to look at the ultrastructure of these boutons, presynaptic boutons, and we could determine that they indeed followed the same gradient of sizes, going from large ones proximally close to the cell body and becoming smaller gradually as you move towards the dendritic tip. And then what we did was to do some more physiology and look at the function of these inputs to basically measure their characteristics in terms of short-term plasticity. So what we did was we patched these neurons and we filled them with a dye so that we could see the dendritic tree using a 2-photon microscope and stimulate either distally or proximally the incoming fibers. What we found was that the distal synapses that are smaller have an increased short-term facilitation compared to the proximal ones. So this means that even if they have a lower release probability initially, the release probability increases with successive stimulation. So they're much more likely to respond to sort of high-frequency bursts. Whereas the proximal ones had sort of a much lower degree of facilitation. So what we found was a gradient in terms of short-term plasticity along the dendrite. Now this also had another really cool implication, that when you move distally, you start getting this increased facilitation, but you also have an effect that depends on the frequency of the incoming stimulus. And what we found is that these distal synapses were very selective to a specific range of frequencies between the 20 and 50 hertz firing rate of the presynaptic neuron, whereas the proximal ones were less selective.
Okay, wow. And what's the next step you think is going to be as a research in your lab?
So to carry on this project, surely we want to understand better how this circuit is organized. So right now we've looked at the single cell and how these synapses are distributed onto the cell. Mm-hmm. We've also found that these basically two different dendritic domains are coding for different types of information, like very selective frequency and non-selective frequency. So we want to know where are these streams of information separate? Are they coming from different kinds of cells? Are they clustered in some way? And if they are, can we recognize these cells in terms of morphology and physiological features? And then to push it even further, we focus mainly on the hippocampus. So is it useful for phenomenon like space coding, navigation, or even working memory and attention, things like that.